Welcome to our presentation on the historical development of the reproductive physiology. This presentation was adapted with permission from the lecture of Dr. John J. Parrish, a professor of reproductive physiology at the University of Wisconsin Madison. Looking back on the historical development of the reproductive physiology in early times, the first writings about reproduction was done by Aristotle, who lived from 384 to 322 BC. He recorded the first recorded idea on how the reproductive system functions was described by Aristotle in his book known as The Generation of Animals. He actually got most of the ideas. Most of his ideas was wrong about reproduction. So, for example, he thought that the fetuses arose from the menstrual blood because during pregnancy, menstruation stopped. Also, he thought that the semen initiated the development of the uh, development when the seminal fluid was deposited in the female during copulation. And he also thought that the semen came from all parts of the body and the testes were only to keep the transporting ducts from getting chinked or plugged with seminal fluid. So all of these findings by Aristotle was, of course, it is not proven and it is not valid during the course of the development of the ideas about reproductive physiology. So moving on to the age of gross anatomy or the Middle Ages. So basically, the age of gross anatomy was the era wherein the scientists are into dissection, dissecting cadavers, looking into different body parts. So the one of the prominent scientists at this age is Fallopius. In the years 1562, he described the oviduct. Thus, we have the term fallopian tube in humans. We also have Coiter in the year 1573. He described the CL or the corpus luteum. And Renier de Graaf in the year 1672 described the antral follicle on the ovary that we often now refer to as the Graafian follicles. Basically, when we say Graafian follicle, it refers to the large tertiary follicle. So we move on to the age of the development of the microscope, and that happened on the 1600s. So the upper left portion here represents the image of the first microscope that was developed by Antoine van Leeuwenhoek. So they have a little lens there that they have used to look through the specimen that they are examining. So at the bottom, we also have the another microscope that have been developed at this age, and as you can see, there is now a little resemblance to that we are using now. So one of the things that they have looked at this microscope is the semen, and they saw these things here. So these are the things that they have seen when they have used this microscope. And one of their conclusion upon seeing this sperm is that the head of the sperm has a little creature in it, which was the developing fetus. So they thought that the head of the sperm contained a creature in there that is the fetus. And they called these things as animalcule. So today, we define animalcule as, we also refer to the animalcule as the little animal. It is an old term now for the microscopic organism. And in this context, it is the animalcule is referring to the sperm at this age. So again, this was the word animalcule was invented by Leeuwenhoek in the 17th century or the 1600s. And the Antoine van Leeuwenhoek was actually the first person with the dubious honor to study or to describe the spermatozoa in detail. So he was a Dutchman who also developed this early compound microscope. So again, the reason why probably they have thought of um, the head of the sperm as having a little curvature is that at this age, of course, now this is the age wherein the microscope is not yet fully developed. So the quality of the image that have been produced might be, of course, now also of poor quality. Next things addressed in the succeeding years was the role of the spermatozoa. So what is the role of the spermatozoa? So one of the scientists that have studied this area is Spallanzani in the year 1780. He was an Italian priest, and he was the first to describe or to conclude that the sperm were the fertilizing agent in the semen. He actually examined a 1 drop of dog semen, and he diluted it with 25 pounds of fluid, and he concluded that it could still be able to fertilize. He performed also the first successful artificial insemination in dogs. We also have Dumas in the year 1825, and Dumas collected oocytes from a rabbit follicles and surmised that animal cues united with this to form the embryo. So he proved that the sperm is a fertilizing agent. So moving on to the age of the modern reproductive physiology, we have here the gonads. So the gonads produce steroid hormones, and at this age, you know, again the gonads has been found out to produce steroid hormones. So for example, for the ovary, you know, we have the estrogen, progesterone, and for the testes, of course, we have the testosterone. Again, this is controlled by the pituitary gland, and they alter, you know, the function of the reproductive tissues. And another is the cyclicity in females. So regulation of the estrous cycle in females occurs repeatedly with a periodic frequency between ovulations. So for example, under this, now we have the use of certain hormones and drugs that will be able to, for example, induce estrus in female animals. Another is the radioimmunoassay. So in radioimmunoassay, this has been used until the year 1995, which uses a radio-labeled antibodies that bind to hormones and detect, you know, the presence of the hormones. Of course, this has now been replaced with ELISA test because, of course, now dealing with radioisotopes is not as safe for humans. Another important, you know, development in the modern reproductive physiology is the use of artificial insemination, and under this, you know, we have to develop an understanding, of course, of the sperm function, leading to the success of artificial insemination. So from artificial insemination, there has been also the development in, for example, the cooling of the semen and the freezing of the semen. So when we freeze semen, we do cryopreservation or semen cryopreservation, also known as the sperm banking or the sperm freezing. Now, this is a procedure to preserve the gametes, such as the sperm cells. So the semen can be used successfully and definitely after the cryopreservation process. Another is the use of prostaglandins to control the estrous cycles. So in the year 1960s, the prostaglandins was discovered, and it was found out that prostaglandins can be used to regulate the length of the estrous cycle. And this can be used, for example, to manipulate the estrous cycle. And we also have the field of biotechnology, and example of the studies or the research under this are for example, the separation of the X and Y sperm. We also have the embryo transfer, embryo freezing, the electronic detection of estrus. We also have the in vitro fertilization, cloning, and others. So these are the forefronts of the reproductive physiology research, in the field of biotechnology. So how do we approach, learning all of these things, in the modern reproductive physiology? So of course, we must begin, by developing a basic knowledge base of how the particular reproductive system works. So for example, when we are developing the cryopreservation process in different domestic animals, of course, we have to understand how the sperm are produced, how is it how it undergoes, certain types of development in the reproductive tract of the male animals. So a series of investigation then will be conducted that can perturb the system. So how do we perturb the system? Now, for example, in the female animal. So we do the perturbation of the female reproductive system, for example, by doing certain types of manipulation of the ovary or the uterus. So, for example, cutting out the ovary or the uterus for the females. For the males, for example, cutting out the testes, and then you give things back. So, for example, you give a hormone to that particular animal, and you see whether you can maintain the characteristic behavior that you remove from that particular animal. So, for example, when you remove the testes, what could happen to the animal? And when you give, for example, a hormone to that particular animal, what will happen due to the response of the animal to that particular hormone administration? Another is the manipulation of the system to improve reproduction. And one of the applications of this is the estrosynchronization. So during estrosynchronization, we basically aim to regulate or manage when to breed the females. So of course, this is important in large-scale production in industrial animal agriculture, because we are dealing with large animals, or a large quantity of animals. So we need, for example, to synchronize the estrus by using hormone preparations in order to, for example, save labor and synchronize, when the mating will occur, when or the conception, as well as the time of calving will occur in a particular farm. So the first area in the application of the reproductive physiology is on enhancing reproduction. So it is said that even a small improvements in the reproduction performance of the animal, can have a profound effects on the uh, animal production across the all sectors. So for example, in beef, in pigs, as well as in dairy animals. So for example, 3% improvement in the birth rate would result in an additional 1 million beef calves per year. And for the swine sector, for example, a 3% improvement in its birth rate would result in a 3.2 million additional pigs per year, and a 3.7 million additional gallons of milk per year, and this is in the United States. So therefore, in the animal reproduction, when we are going to improve the animal production performance by even a very small amount, such as 3%, there could be a profound effect, to the economic output of these animals, because it has a large effect on the efficiency of these animals in terms of producing offspring, as well as in producing, for example, milk for the public consumption. Let's now proceed to the current trends in animal reproduction. So animal scientists are currently working to increase reproduction, as shown in this diagram. So there is an upward vertical arrow there pointing upward. So that means that the the field of animal reproduction is working to increase the production performance of animals. So when we say production performance, we are referring to, for example, the growth of animals, as well as in dairy, for example, in dairy animals, and the milk production of these animals, and many other production parameters. So what it will do is it will cause changes in the metabolic as well as in the physiological state of the animals. And when this happens, it could result to a somehow decrease in the reproduction. So when we are focusing more on the production aspect, we tend to increase the production performance. There could be changes in the metabolic aspect of the animal, the physiologic state of the animal, that will decrease, most likely, the reproduction. So when we do this, so we actually most likely encounter problems with reproduction. Hence, there is always a continuing need to improve the reproduction or the reproductive performance, and also on how on the understanding on how we'll be able to apply new technology to this aspect. So the second area of the application of this concept of the animal um, reproductive physiology is on limiting the reproduction. So when we are referring to the limiting the reproduction, we tend to think of this on humans. So most likely, we likely first think of this concept with respect to humans. So this is true, because as the world's population increases, there will also be an increased need to control reproduction so that the resources are not depleted and pollution does not make the planet uninhabitable. So in the case of pets, we control reproduction by, for example, spaying or neutering dogs and cats. And these are, of course, cheaper ways of accomplishing this, and will always be of interest to the animal reproduction sector. So we limit reproduction in our pets through this procedure, spaying and neutering. In the case of insects, the population control of insects will likely involve the control of the reproduction, as chemical toxins continue to have damaging effects to humans. So, of course, insects could be beneficial and harmful. Now, for those insects that are harmful, we tend to control these insects because, again, they serve as pests to our crops. So we tend to control them by chemical means. And when we use chemicals, of course, this is toxic, not only to humans, to animals, but also to the environment. So the main goal here now is to use alternative procedures, alternative control measures in order to control the population of the harmful or the damaging insects. In the case of the wildlife, a good example of this is for example, the deer population. It needs to be limited in the agricultural areas to prevent crop damage. And this is somehow true also with the insects. There are certain wildlife species that when they are increased in terms of the population, they can serve as pests to humans and to the agricultural plants or the agricultural crops. Of course, in humans, again, the increase in population would be not beneficial. It would be damaging to the resources of the available resources of the state, and of course it can also cause the pollution, because when there is an increase in population, there is also an increase in the amount of the waste that are being produced, and of course this can have a harmful effect to the environment and to other animals as well. So in humans, of course, the main goal of certain nations is to decrease, no, the population.